the next speaker is Yossi Yovel from Tel Aviv University. So he's an expert in bats, and he did his sorry. So he did his bachelor and master's degree at Tel Aviv University, and his PhD at Tübingen at MPI, and then joined as a professor at Tel Aviv University. So I should say that once walking in the Ben Gurion airport in Israel, I've seen his face appearing on the wall. Basically, there was an exposition that showcases some research and science done in Israel, and he was one of the people featured there. So I guess in the interest of time, the floor is yours.
Thanks, Michael. Thanks for inviting me. Okay, you can see it now in presentation mode, right? You can see my my presentation? Yep. Okay, great. So hi everybody. I'm going to talk a lot about bad communication, but before bat, I should say, not bad. In Germany, sometimes they mispronounce TSD, and I was once presented as the student of a very famous bad researcher, but the word is bat. Before I talk about communication, I want to tell you a few words about bat sociality, really just one slide. So what are bats actually talking about? So here's one example. Hopefully the movies will play. So apparently there are different types of bats in a fruit bat colony. For instance, if you place a bowl of fruit in the center of the colony, so now on the left, hopefully the movie hopefully you can see it. The bat lands on the bowl of fruit and takes some food and flies with it to the ceiling. We call this a producer. The term was invented mostly by bird researchers. We did not invent these terms. But something like 30% of the individuals in the colony will never go down to the bowl of food. They would prefer to go to some other specific individual and scrounge food directly from its mouth. And I already gave you the answer when we looked at these preferences, and this is the network down here. This is what it represents. We found that these are specific preferences. So this is a producing scrounging network where, for example, this arrow means that this individual prefers significantly to scrounge food over months from this individual. We later on analyzed, so you can see that there are two colors here, and I can tell you that the colors are females and males. And we later on also found that many of these ties, not all of them, are related to mating. So the females seem to be testing the males by scrounging food from them. This is, of course, an interpretation. And at the end of the season, of the mating season, they mate with one of these males, probably the one with which they created the strongest bond, although, of course, this is my human interpretation. Now, many of these interactions, we're still talking about fruit bats. You'll see several types of, several species of bats in this talk. Many of these interactions are accompanied by sound. So here's an example. I don't know if you can hear me. Can you hear me? Okay, so I think you could not hear me while the movie was playing, but what you saw, okay, thanks, great, were interactions between bats in our colony after foraging and before falling asleep. At the end, you could see them clustered sleeping. This is their nightly cluster, or their daily cluster, I should say. And they can spend hours in these arrangement fights or whatever. And you could hear that there's a lot of communication going on. And now I want to take you into a bat colony. I'll say this before you see the movie. A bat colony with thousands of individuals, still fruit bats, and see what it, or hear or listen to what it sounds like. Okay, so here it is. Okay, so the first question that I want to present, the first question that we studied is what kind of information is embedded in these agonistic aggressive calls. Okay, if you enter a fruit bat colony, it sounds like they're constantly fighting with each other. It seems meaningless, to invest so much energy in vocalization without it conveying some information. By the way, you can see the students for each of the studies. You can see the students who carried out all of the research. I can tell you, by the way, that if I train you quite quickly, you will also be able to distinguish some of the vocalizations in this cacophony. So I can hear the pups, for example, but we will come back to this later on. So of course, recording bats in this environment would be possible, but the data would be impossible for us. So we had to go into a more controlled setup. Before I show you the setup, I just want to show you what the calls look like. So these are spectrograms. Okay, we saw spectrograms yesterday, but just in case, it's a representation of sound. We have time on the x-axis and frequency on the y-axis, and energy is depicted by color. And you can see here the repertoire, or part of the repertoire of fruit bats. So these are mating vocalizations emitted by a male while mating. These are isolation calls emitted by, or distress calls emitted by a pup. We will come back to them later. This is very typical to a very angry individual. These are echolocation calls. We'll come back to them later too. But the big majority of vocalizations, at least in the colony, look like this. Okay, so they are aggressive calls. And this is the corpus of recordings that we are focusing on this study. We were wondering whether we can extract more information than just saying these are aggressive vocalizations. Okay, so this is what the setup looks like. We actually created many small bat colonies. We divided the colony into two parts. It's very small, but still we divide it into where they sleep and where they feed. And the idea is basically to get very high quality, a lot of data. You'll see in a second how much in very high quality. Everything is covered with foam. So the recordings have very few echoes. We're recording around the clock. We're recording everything basically for several months. And this gives us a database originally with millions of vocalizations. But after we filter them to good vocalizations, we go down to hundreds of thousands. And after we annotate them, okay, so for each interaction, we detect who is the emitter, who is the addressee, what was the context. In a second, I'll come back to what does this mean, what does context mean. And only for these 20,000 vocalizations, we then run our study. It's still a big database, but much smaller, than the one we started with. But it's annotated, and of course you can see this huge reduction. Maybe you're wondering why is it such a huge reduction. The reason is that if we're sure about this information, do we count this as an annotated vocalization? So we just throw out everything we're not sure about. We can still use this, right, for unsupervised algorithms, for example, like Michal just discussed. So I want to show you what an interaction looks like in one of these small colonies. It's all with the security cameras because we can't create. Anyway, we're creating tons of data, so we're trying to limit this. So this is what the interaction looks like. Hopefully the movie will play. Okay, this movie is always a bit problematic, but what I want to remind you is that the bats actually experience the same interaction like this, right? Because they're perching in the dark, it's mostly dark, and they see this or feel this or perceive this as something like this. Okay, so what we're actually asking in this study is whether when, sorry, my presentation is stuck. Okay, whether when hearing such an interaction, a third bat that is listening, whether it can say something about who is the emitter, who is the addressee, what is the context, I'll come back to this in a second, and also maybe something about the outcome, what is going to be the outcome of the interaction. Okay, so what do I mean by context? I mentioned this several times. Obviously, and this is a huge limitation, something I'm willing to discuss later on, we can only define context that we can see with our eyes. Okay, so that's our human, at least with this type of supervised approach, this is our human bias. And we detected 4 types of interactions. So they're all aggressive, I remind you, or we at least see them as aggressive. So you have interactions over food. Okay, they look like this. Okay, so one individual had a piece of fruit, another one was trying to scrounge. These are mating aggressive interactions, but this is not what you've seen before. In this case, the female is kind of aggressive towards the male that is trying to mate. Okay, we have fighting with no clear reason that we can detect. And probably the favorite of my favorite at least is the sleeping interactions, because it's, to me, so human. They're sleeping in a cluster, one individual wakes up, it's moving the other one, and the other one is shouting at it or something like that. Okay, so now you understand the data. In terms of the analysis, we're using a supervised algorithm. We're not using a neural network, although we tried neural networks as well. The data is presented as Mel capture coefficients. We can discuss this later. We tested many different representations. It didn't really change the results much. And our classifier is a Gaussian mixture model, or a type of Gaussian mixture model, which is known as a GMM-UBM. It has some kind of a background model, which is very common in speech processing. And as I said, we tried other approaches. The results were quite similar. What I want to emphasize here is that, for those of you who don't know, the fact that we're using capsual coefficient means that we're just looking at the frequency. We're not looking at temporal information at all, although I think it is very important. So this is probably an underestimate to what bats can do. And here are the results, okay? So graphically, they're quite boring, and we're looking at the confusion matrices. So basically what this means is we have 7 individuals here. So this is for taking a vocalization and predicting the emitter. So we're looking at 7 bats for which we had most data. And this is our prediction, and this is the true class. And if the diagonal, okay, so this means that we're right 80%, for example, for bat number 3, more or less. Okay, it's the balanced accuracy for those of you who know what that means. But basically, if the diagonal is yellow, it means that we are doing well. So we can take one of these aggressive vocalizations and say quite accurately who is the emitter, okay, based on frequency.
Sorry for interrupting. Can I ask a quick question? Where do we have the ground truth about the emitter? How do we find?
Yeah. So this, if you remember that huge reduction from 160,000 to 20,000, that is because we manually observe the videos. We now have an automatic method to do this, but we manually observe the films. We have the videos. We have multiple students doing this. We do cross-validation, and we throw out everything that we're not sure about. Okay, so it took a huge amount of time just to annotate this data set. Very hard to do automatically. So, you know, if I could, for example, maybe detect the emitter automatically, but detecting the context would be probably impossible. And this is another explanation, maybe I didn't say this, why we had to reduce the size of our colony to small colonies. Okay, if I had 20 bats there, I would not be able to say who is the emitter. Okay, today we do have a method to do this, but still I would lose a lot of information.
Of course, would you know from one day to the next that this is the same bat, or is it all within the same?
Yeah. No, they're all marked. Of course, they're all marked. We know over 3 months that these are the same bats. Of course. Yeah. As you can see, I can also do well above chance in predicting what is the context. So out of these 4 contexts that I've described earlier on, so I can take a vocalization and say whether the bats were fighting over sex, over food, over sleep, or without a reason that I can see. And interestingly, we also can say something, again, everything is compared to chance, so there is some information, even if it's far from perfect, about the addressee. Okay, this is something that surprised us, but I can say whether a female or a male is being approached, and to some extent, I can also take all of the females, for example, that have been approached and say, above chance level significantly, which female is being addressed. You know, if we think of humans and we anthropomorphize this problem, then this makes some sense, right? When I approach one individual using the same vocalization, it might sound differently than when I approach, you know, my boss versus a friend, for example. And also, to some extent, we could say something about the outcome of the vocalization. So we define 2 outcomes, whether the bats stay together or whether they separate after this aggressive interaction. So this is actually these are the conclusions of the first project that I wanted to present that has to do with information conveyed by this aggressive vocalization. All of this I've already said. It's a single vocalization that we use, and we're using spectrum or spectral features only. The next story that I want to tell you has to do with the ontogeny of vocal communication. So this was mentioned yesterday already regarding the elephants. There's a big question about whether animals, and mammals specifically, learn their vocalization, their vocal repertoire, or whether it is innate. And basically, there are very few mammalian models. Of course, songbirds are very known for their ability to learn vocalizations, but there are very few mammalian models for which it has been robustly shown that they learn the vocalizations. Now, and we wanted to test this with bats. There were a few previous publications with other species of bats suggesting that they might be learning. And what I'm showing you here is just an image representation of the ontogeny. So all vocalizations start with this, what I called an isolation or a distress call that pups, you could think of this as crying, basically. So from day 0, when you take a pup, you move it, you put it alone. This is the vocalization that it will emit. It's rather high in pitch. I will play it back to you now, but it's high in pitch and not very loud, so I'm not sure you will hear it. I'll try. Okay, I don't know if you heard this, but when I said it in the cacophony, I can hear the pups crying. This I can hear. My ears still allow me, and you can see how over a period of almost a year. Okay, so this is day 150. These vocalizations turn into this adult-like sequence that we've seen before. Okay, and the question is whether pups need to hear adults in order to evolve this Okay, and in order to do this, in order to study this, there are several classical ways in the literature. One is to perform an isolation experiment in which you deprive the pups from the ability to hear adults, and that's exactly what we did. So we had pups isolated with their mothers. Okay, so the pup was with its mother, which is important for the development of the pup, but luckily the pup and the mother hardly communicate. And we of course, we controlled for this, we checked this. And the reason is that the pup is under the mother's wing, okay? And it's connected to the nipple, and therefore there's hardly any interaction. So the pup actually mostly listens to the adult interactions. This is something I'll come back to in the next story. And you see, what you can see here is over time, okay, so this is day 210. I'm comparing, and this is for so we extracted different acoustic features. And if I look at all acoustic features, this is some kind of an average, and I compare them to the adults. So you can see that the control pups, the ones that were exposed to adults, okay, to adult communication, converge quickly or within 100 days to the adults, while the isolated pups stay or lag far behind. Okay, it seems that in the end they might be converging. We're not exactly sure what's going on here, but definitely you can say that there is a very substantial delay in their vocal ontogeny, which suggests that there is some role of learning. You can see this here for the pitch or the fundamental frequency actually only. Okay, so for a specific vocal feature, okay? You can see the same pattern. Now okay, so there is some kind of lag, but what's going on here? We're trying to say something more conclusive about what's going on here. So hopefully this slide says a little bit more about where is this delay coming from. What I'm showing you here over time, okay, you can see day 0 to 70, 70 to 140, 140 to 240, and we are now looking at a 2 feature space. Okay, so we're looking at 2 acoustic features, the fundamental frequency and Wiener entropy. You can plot others, of course. The black area is the adult region. So adult vocalizations will be within this region. And you can see how the control pups, they start all over the place, but somehow, as if they are guided, they converge quite quickly onto the adult range. But if you look at the pup vocalizations, you can see that the majority of vocalizations converge to the right area, but you still have a lot of this, it's really dispersed still. So you still have a lot of vocalizations that are outside the adult range. So it's as if they are lacking this guidance. It's as if hearing adults guides you which vocalizations to use. It's not that they cannot emit adult-like vocalizations, but more that they need this guidance in order to converge onto the right regime. Okay, so it seems to be a mixture of innate, some innate components, but also there is some kind of learning. Now, of course, when you isolate the pups, I already hinted on this. Some people could say, okay, these pups have no mothers, and this affects. They have their mother, but they don't have any. They don't observe any social interactions. So maybe it's not vocal learning, but something is wrong with their sociality. So this is why we came up with the following experiment. So it's a different study, and what we did here is to have both. So we have 3 groups of pups, control and 2 manipulations, And all groups of pups are with their mothers in a group. So we have 6 pups and mothers, normal colony, small colony, but normal colony. They're exposed to adult interactions. So they hear their parents or mostly their mothers. But in addition to hearing their mothers, we also manipulate them by placing speakers in the colony and playing back, if you wish, a dialect. It's hard to say whether it's an accent or a dialect, right? Because we don't know if these are words or what exactly these vocalizations will represent. But we play back a subsample of the vocal repertoire. So you can see this here along the fundamental frequency. You can see the control. So this is the normal distribution of fundamental frequencies. Green is the control group. This is what they hear. Blue is the low pitch group, and red is the high pitch group. This is what they heard. So I always say, I always, when I give a public talk, I say it's like taking a family and moving from one city to another, from London to Glasgow, for example. So the children are exposed to both the dialect of their parents at home, but also to the new dialect where they moved to. And there are many analyses to show. I just want to show one slide that summarizes this. What you see here are linear discriminants, or you could think of this as principal components, basically. And we're plotting, and this is time. Okay, so again, you have the days. So this is 1 year. Okay, and you can see the 3 groups. You can see that to begin with, the 3 groups are all together, more or less, red, green, and blue. And you can see how over time they split into 3 groups. And importantly, it's not as if we are running a discriminant analysis on the data of the pups, right? Because then we would find some separation. What we've done here is to run the discriminant analysis on the adults, okay, on the playback, sorry, not on the adults, but on the playback of the adults that they were exposed to. So these axes represent the playback, the manipulation, and you can see that on this, in this space, the pups, so we don't do anything, the pups separate into these 3 groups, suggesting once again that there is a role of learning in addition to some innate role, some innate component in their acquisition of their vocal repertoire. We call this a crowd vocal learning, okay, because most studies on vocal learning, mostly in birds, but not only, they talk about a tutor, so a one-to-one interaction and how important this is, even with humans. Okay, this is apparently very important. But in this case, we're talking about learning from the group, basically, right? Because we have this playback. So the pups actually are even though they hear their mothers, the group or the cacophony that they hear in the background influences their vocal ontogeny. And if you think of this movie that I showed you earlier, this is probably not surprising. So imagine a pup under its mother's wing in one of these beds, and this is what it's exposed to. So it's hard to imagine how it can really learn from its mother, at least in this colony setting. Okay, if you have any questions, this is a good time. Maybe if there's any question from the Q&A, wow, it's long, so maybe we'll postpone it. 1 second, I'm looking. No, this is for Michal. I will not answer Michal's questions.
You have one question in the bottom.
Yeah, I'm reading it. I'm stopping now to answer because the next 2 studies that I will show you are completely different. I now kind of finished talking about vocal learning and communication in the lab, and now I'm going to take you out to the field. So sorry, I just read the question. Yeah, okay, good. So the question is whether we looked at it's a question that is often asked. It's regarding the first study where I showed you that there is some kind of information. Our classifier was able to say something about what is the context, for example, but we're using these MFCCs. It's not very intuitive. And the question is which features allow distinguishing the different contexts, and maybe, I don't know, how does the distribution look like? That's my interpretation of the question. So the answer is yes. We also ran the same study by doing the feature analysis, the one that I presented in the second and third study. But if you look at the distributions, they don't cluster very nicely, as sometimes you see with songbirds, for example. Maybe this is an important point to talk about. 1 second, for some reason my presentation is not going backwards. Okay, so if you look at these vocalizations, they're very noisy. Actually, they're reminiscent of human speech. They're higher in pitch, but they're similar in their noisiness. Okay, these are not easy tonal vocalizations that you can very easily classify, and that's a big problem that we have. So they don't cluster very nicely into any group, any clear group. When you do run some kind of a T-SNE analysis or something like that, and you look at context, I have this slide later, very later on, and I don't want us to break the presentation. You do see some kind of separation in space, okay? But it doesn't cluster very nicely. It looks like a big continuum, which is something that we've been thinking about a lot. So for example, if you want to talk about syntax, for example, you need some kind of clustering, okay? And that's something that, which I think is very general for many animal vocalizations. You lack this ability to cluster them nicely. Okay, so since I still have some time, I'm going to move a little bit from communication and show you how we study these animals in the field and how we plan to connect this to communication. So everything I showed you so far was in the lab. Okay, even though I always advocate for studying animals in the field, and that was due to our inability to record them nicely in the field. I think I explained why. But in the past several years, we've been developing the ability to record animals in the field, including their vocalizations. Okay, so what you can see here is a miniature device that we ourselves developed. It includes a GPS. You can see here the movement of bats. I will not be talking about these bats today. These are nectar-feeding bats that fly hundreds of kilometers every night. Okay, you can see GPS data over here. And we have a microphone on this setup. This is the microphone actually. And in a second, I'll show you what kind of data we get from the microphone. Obviously, we get vocal communication, but we also can do more. We have an accelerometer. You can see here the wing beat of the animal in red. We have physiology sensors. So you can see here, for example, ECG data recorded on board the animal. So you can basically record many types of, you can also connect a camera, but it makes everything much more difficult. But we can record many types of information. Now, I said that the microphone gives us a lot of information. Here is what I mean. You have to remember that most bats echolocate, which means that they use sound in order to sense the world. Okay, and I'll show you later on soon how these sensory vocalizations can also be used for communication by the bats. Okay, this is why I'm talking about this in this meeting. So what do we see here? So we see spectrograms once again, and these are echolocation calls. Okay, you can see they're multi-harmonic in this case, in this bat. You can see that they're ultrasonic. So this is 25 kilohertz more or less. And note that I can study interactions between bats, because when I have a microphone on the bat, I can also record its conspicuous. These are nearby bats. Okay, so I can study interactions, for example. I can study sensory interference, for example. I know based on sound only when a bat is attacking prey. Okay, so what you see here is a feeding buzz. It's very important. I'll come back to this later on. You see that the calls, there are many things here that change. You see the intervals are becoming shorter, the calls are becoming weaker and shorter themselves. This means that the bat is attacking food. And I can see this even when the bat is flying, 1,000 meters above ground or somewhere, wherever it is, right above the desert in the border of Mexico and between Mexico and the US. Sometimes I know what is being attacked. So for example, this is the frog-eating bat in Panama. And the frog is also emitting sound, vocalizations, okay? I can record them. And I know that the tundra frog has been attacked, and which is one of the favorite prey types of this bat. So the bat itself, by the way, is also using these vocalizations in order to find the prey. And in this case, I even know that this was the last call emitted by the frog, and this is based on the chewing sounds right after the attack. So I even know to some extent whether the attack was successful or not. So I can do all of this using sound. This is actually one of the reasons why I claim that studying bats in the wild has huge advantages. Try to extract this information for an animal without observing it. And one of the questions that we studied using this system is indeed sociality and foraging socially. I will describe the movie before you see it, because again, I'm not sure you will hear me. So in a second, I'll play this movie and you will see thousands of, they're called greater mouse-tail bats. You can see them already here aggregating at the exit of the cave. And you'll see them flying out, you will hear them. What you will hear is social vocalization. There's a lot of social communication going on before they emerge, so you can't hear the echolocation. And then the camera will turn and you could see for a second, you'll see the Lake of Galilee. So these bats are flying towards the Lake of Galilee. Okay, I hope you could see I was pointing towards the Lake of Galilee. So, and I forgot to say, this is just before sunset. Oh, sorry, just after sunset. Okay, so it's there's still light, but it's just after sunset. You could see a kestrel, a predator, trying to get one of the bats. I couldn't speak during the movie. And I like to think that 2,000 years ago, Jesus was walking on the Lake of Galilee, and he looked up and saw this swarm of bats flying above him. And then he probably asked himself two questions. Okay, so the first one is, how do they avoid sensor interference? And I'm not going to talk about this today. It's a huge problem. They're all emitting calls at the same frequencies. We have several studies published about this one, very, very recent. And the second problem that I want to talk about, the second question, is, are they operating together, these bats? Are they doing something together? And I'll give you the answer already. The answer is, we believe they are. And here is why. So this is the bat, by the way, greater mouse-tail bat, 35 grams, one of the smallest animals ever tracked using GPS. And you can see that this animal, so during summer, this animal searches for queen ants that are flying, so performing a nuptial flight. So it's a very difficult search. And you can see it here, okay? So this is the movement, look in green and blue, look at the scale. It moves dozens of kilometers every night. It moves, so blue is one night, green is another night. So it's a very exhaustive search. So we call this a search for ephemeral food. Okay, you have to move very, very far. There's no way to predict where food is going to be, and you have to search a lot. And without going into the details, what we show is that the bats are constantly moving in a group. Okay, so it's not only that they emerge as a group, they are constantly moving in a group. Okay, this is something about the density of the bats, but really, instead of going into details, I'll just show you an example. So this is one individual. Okay, it's the same blue trajectory over here, and I overlaid in a red flag. You can see wherever the bat is attacking food, and in green you can see wherever the bat is flying near other bats. Okay, so it's constantly flying near other bats. I should say our microphone is much less sensitive than the bat. So if I don't see a bat nearby, it doesn't mean that there is no bat. Okay? So this is an estimate, and you can still see that the bats are constantly moving together. We see this for several bats. Okay, this is one of the most fascinating bats that I know. It's called the Mexican fish-eating bat. It roosts on these islands in the Sea of Cortez between Baja California and Mexico, and it has this very difficult task, very similar to the other bat I showed you. It has to fly off the island. These are our GPS tracks. For the first time, we reveal what these bats do. And it has to fly dozens of kilometers in search of, once again, ephemeral prey that is hard to predict. It fishes, it eats fish or crustaceans on the island, on the ocean, sorry. And it doesn't dive in, by the way, okay? It collects them with its claws. And I'll show you now a movie, and I don't know if you'll hear me, so I'll say already now what you're going to see. And you'll see bats flying off the island. It's going to be several hours per bat, but shortened, compressed in time. They fly back, and you will see two circles, white circles whenever there's a nearby bat, and red circles whenever they're feeding. And note that how they are constantly moving with other bats nearby. Can you hear me now? Yes, okay, so this movie is silent, so in this case you can hear me. So you could see, for example, here the bat found a patch or a school of fish or something like this, and it's foraging. But more important for this talk are these white circles, okay, suggesting that also when they move between foraging patches, they are always moving in a group. Okay, so the interpretation, the conclusion is, or let's say the hypothesis is, the hypothesis is, okay, that they're searching, that somehow moving in a group helps them, and we actually prove this by using a simulation. Of course, it's hard to prove analytically, but the idea is basically that if the food is patchily distributed, so this is a schematic suggesting what's going on. If there's a lot of food and you move in a group, this is beneficial. Why? Because each individual bat can search a very small range. Okay, using echolocation, you can only see a few meters ahead. So I always say it's like driving on the highway using a small flashlight. It's very difficult, but you can eavesdrop on other individuals. Okay, this is this huge range, and you can hear other individuals from more than 100 meters, more than 100 meters, sorry. And you know when others find food. How? I showed you this this feeding buzz earlier on, right? A few slides ago. So when somebody else finds food, you know this if you can eavesdrop on it. This is why this bag of chips appears here, because we always said it's like a bag of chips. If somebody opens a bag of chips in a dark theater, everybody knows there is food over there. So I want to, in addition, to explaining this, and we actually simulated too. Here's a simulation of this using a very, I hope you can hear me, but it's a very typical, simple, what's known as a 3-radi collective movement model. And you can see that using this same schematic, the same idea that I showed you earlier on, the bats form groups, and there's some food over here. And indeed, we show that using this approach, they will find food faster than when moving alone. But in addition to showing why collective movement assists them, I want to point out again to this message that there is no real vocal communication going on. Even though these bats have vocal communication, they communicate a lot, as we heard in the cave, for example. While flying, they don't need this. They know what the others are doing just by eavesdropping on their echolocation signals. Okay, hopefully you could hear me during the movie. If not, somebody say something. The last thing I want to show you, I'm going to take you completely out of the communication world and talk about different types of cognition and how we can use this technology in order to study cognition. And I'm going to talk about cognitive maps. It will take me a few minutes only, hopefully. And just in case you don't know what a cognitive map is, the idea is that you have, and it's highly debated, which animals, including humans, have cognitive maps. But the idea is that that if you have a cognitive map, you contain, or not the wrong word, but you have some kind of a mental map of the environment in your brain. You hold it in your brain. Okay, maybe that's a better word. And how do I know if somebody has a cognitive map? So the hallmark, or one way to test this, is to show that you can create shortcuts. Okay, so if you look at this bat, we track this bat in the wild for almost 100 nights, and look how far it moves. So it flies 25 kilometers to point A for many nights, and then it flies to point B, and then it flies to point C for many nights, and then it performs something that looks like a shortcut. Okay, but the idea why I like this example is that this still doesn't prove, or even doesn't hint towards a cognitive map. And that is because in order to prove a map or to suggest a map, you need to show that this is a novel shortcut, right? It could be that this bat performed this, maybe it was flying with a map, right? Maybe it was flying with GPS a few years earlier, where maybe it was flying behind its mother. Okay, so we can't say that this is novel because we don't know the history of the animal. Okay, by the way, these animals can live up to 40 years. So the history is a long history. So in order to overcome this challenge, we invented the following idea. We created our own colony. So our bats roost in Tel Aviv University, but they are free. They can do whatever they want. They fly out and come back. Not all come back, by the way. And this allows us now to monitor the same animal from the day it is born. Okay, so I can show you now, I'm showing you now, sorry, 3 nights of an adult, not a newborn in our pup. And you can see nicely how this individual, for example, so each color is a different night. And you can see how this individual flies over, it finds another colony, also very interesting for me, sleeps over at friends, and then comes back and sleeps in our colony. And similarly, we will have bats from this colony coming and sleeping over in our colony. So our colony is really part of the network of colonies in the environment. And now I'll show you an individual. I think it's something like 60 nights, one individual. You'll see all of its movements. Again, I'm describing the movie in case you can't hear me. You will see every color is a different night. You will see how it increases its foraging and exploration range from blue to red. So it's the same bat over something like 60 nights from day 0, from the first night outside. Okay, so the nights are moving from blue to red. See how there are a lot of nights where it goes back to familiar locations, but there are also nights where it explores. Right? And I should say day 0 is the first night of this bat outside, but it's not its first night in life. Okay, so this is something around day 40 or 40 to 60 for this bat. But now when I see the bat flying over here and coming back, so I look at the yellow precedes the red, I know it has never ever been in this area, and still it is able to come back in a rather direct line. Okay, so I can now look at ontogeny of navigation. You can see how the bats increase their exploration. I already showed you this. The good thing about these bats is that they are fruit bats, that unlike the fish-eating bats that I showed you earlier on, they go back to the same trees again and again. So when I see a bat, so we mapped more than 1,000 trees used by our bats, and when I see a bat arriving at the tree, from a familiar tree, I can guess, imagine, hypothesize that that's where it was aiming for. Unlike with fish-eating bats, where you never know what the bat is aiming to do. And now I'm going to give you a very, very brief overview of the results. Basically, we show that the bats have the ability to perform shortcuts, and indeed novel shortcuts. So you can see here an example of a shortcut in blue. Pink is what the bat performed before during the same night before the shortcut. This is some kind of a foraging tree, and then you can see the shortcut. We have shortcuts up to 20 kilometers. This is 500 meters, but it can be much longer. And I know this is novel because in white you can see everything the bat did before this. Okay, so I have the full history of the bat. We detect hundreds of shortcuts, or actually slightly more than 200 shortcuts. And here are just a few examples. I'm not going to it, but we also show that these are probably based on vision. So although these bats use echolocation is a short-range sensory system, and when navigating like this, they are probably using vision. A big question was, from what age can they perform shortcuts, and what day outside? And we find that it doesn't change over time. So you can see this here, the rate of shortcutting over time, and it seems to be completely uniform, maybe except for the first few days. So the bats have the ability to perform shortcuts from day 0 outside, again, not day 0 in life. But if you look at the overall distance of the shortcuts, you can see that this is increasing. Okay, so obviously, the more they map the environment, over here, for example, the longer the shortcuts can be, right? But they can perform shortcuts. If this is, for example, a very early day, they can perform these shortcuts from day 0. And the last thing I want to say is, okay, so how do they learn the environment? And this is a whole new study, unpublished yet, but recently submitted, in which we look at the interaction between mothers and pups during this ontogeny of navigation. And the thing is, I'm not sure how many of you know this, that in this species, at least on all species, mothers fly with their bats attached to them, at least for a long period. And we actually know that the bat is attached to the mother because we have an accelerometer on the mother and on the bat. We have a GPS, but also an accelerometer. And you can see here the acceleration of the mother and the pup, and you can see how they are perfectly, synchronized with a phase shift, right? Because the mother is flapping, is going up and down. The accelerometer on the pup is reversed, and therefore you see this antiphase. But they're perfectly synchronized, so we know exactly when the pup is on the mother, like over here, and when it is not on the mother. So here the mother is flying, but the pup is stationary. And what we find is that the mothers place the bats from a certain age. They fly out with them, but they then place them on certain trees within a radius of up to 7 kilometers from the roost. And then later on in life, after a few weeks, the pups will start flying independently to these trees specifically. So they seem to be learning from the mothers, even though they never flew independently. They're always carried on by the mothers. So they are learning passively while they are being carried on by the mothers. So I finished. I finished a little bit early, I think. So that's good. You have your break. We can all go out and drink our coffee together in the corridor. And I just have to thank a lot of students who collected this data, some of which I mentioned already, and a lot of collaborators and funding agencies. And that's it. Thank you very much. I'll be glad to answer questions.
Thank you very much. Really wonderful talk. Very exciting, and well, it seems that we have a lot to learn from the bats. So I think there are several questions in the Q&A and also in the main chat.
I think they're in the chat.
Yeah, in the Q&A, I don't see anything new. Let me know if something appears. And in the chat, no, this is the Q&A. How do I open the chat? I saw it a second ago. There you go. Okay. Let's see. Where are my questions?
Starts from Luigi.
I think starting with, have you been able to track the path of individual learning? That's me, or that's earlier on the time?
2 down.
That's me. 2 down?
Yeah.
Ah, okay, great. Ah, Luigi, okay. For food, the decision and see. Yeah, if I understand the question, how do they count the agreement to follow? Okay, great. So if I understood the question, this is relating to the collective movement, where I showed you the fish-eating bats, basically. And I was quick there. I didn't know I will have so much time left over, but the idea, at least how we imagine it and how we modeled it, and it works, is that there is no communication. So nobody shouts, "I found food." Okay, I don't know if I have time to go back to the movie. I will not do it, but I'll be happy to supply it to anyone. So nobody's shouting there is food, and the only guys who know that food was found are the ones that are eavesdropping on the ones that already found the food. Okay, so basically only know what the bats within 100 meters for you are doing, but still information propagates through this group. I call it, I like to think of it as some kind of an avalanche of information. So the first individual finds food, and then the ones around it find the food, and the ones next come closer because they're moving in this direction, and they find the food too. Obviously, not everybody finds the food. So if you paid attention to the example that I showed, you could see that that a part of the group actually diverged away from the food. Okay, so it's definitely not an optimal solution, but the nice thing is that it's very, very cheap on communication. So there's no leadership, there's no direct communication. You just eavesdrop, and you still perform much better than as an individual. Yeah, so I think the next question is related. Do they have a collective memory not to throw for food in the same location? Yeah, it's a fantastic question. I haven't tested it. I imagine that you could look at the movement of the of an individual. You have to remember one of our caveats is always we track a few individuals within the group, within the swarm, and we have to from this to deduce what's the collective doing. I'm thinking now out loud, and maybe you can help me whether you could use the movement of the group, of the individual, sorry, and say something about whether the memory is not individual but collective. But definitely you can see a movement that suggests that they're not going back to places where they've been before, but I have to think about it a little bit more. Assuming they can map, sorry, oops, no, I jumped down, sorry. Assuming they can map an environment and learn from others, do they expect symbolic learning? Yeah, I would love to show that. Of course, our dream is to find the bee dance for bats. We have no evidence for this so far. Okay, so we don't have any evidence that they really provide information. And unlike social insects, you have to remember that these are basically selfish. It's a collective of selfish individuals. So we don't have any evidence for this. But obviously, this is something we would like to study. Definitely, we hypothesize that the mothers are investing energy in order to teach their pups, but I don't think that vocalizations are involved. By the way, there is the vocalization, a mother-pup vocalization, something I think somebody mentioned yesterday. So when a mother is searching for its pup, she left it on a tree and she's coming back, for example, and she doesn't find it, she will emit a very specific vocalization that sounds like the isolation call. You remember those isolation calls that I played back earlier? So there are adult calls that sound similar, and the pup will start responding to these vocalizations. Whether it is specific, whether the pup recognizes its mother and responds only to it, I don't know. but it's actually heartbreaking to see a mother, I've seen it, searching for its pup and shouting these vocalizations, but without any response, or sometimes, hopefully most of the time she does get a response. Yeah, okay, thanks Joyce for the comment. I think it's very interesting to compare, by the way, although the bats are probably less working as a group than the elephants, but in both cases it's a mammal with long-term memory. The fruit bats also memorize an array of different feeding sites. So all of this, I think, are commonalities that are really interesting to compare. Increasing is not flight. Yeah, if you look at the adults, so there is a study coming from the Hebrew University that came out together with our study recently, like a few weeks ago, and we looked at the pups, they looked at adults, and it seems as if the adults are more stereotypical in their movements. So I think that this exploration, a lot of exploration that we see decreases over time, just like you suggest, yes. Do you hear Egyptian fruit bat vocalizing? When implied. Okay, so except for this mother vocalization that I've just mentioned, the answer is no. And in general, we recorded many species, more or less.
Sorry, Yossi, can you repeat the question? Not everybody can see the questions.
Yeah, sorry. The question was, do you hear, I think I read it actually. Do you hear it? Maybe I read it quickly. Do you hear Egyptian fruit bats vocalizing when in flight? So I said, except for these mother cries that I mentioned a few seconds ago, the answer is no. And actually, for most bat species that we've observed, we don't see any social communication in flight, which I find very interesting. In the case of the fish-eating bats, it seems as if they are sometimes vocalizing, but mostly near the island, and not so much when they are out at sea. The last question, I'll read it and then I'll shorten it probably. Yeah, okay, so the question, this goes back to the second study I showed on vocal ontogeny, where I showed you that that if you remember, the isolated pups were much more dispersed in acoustic feature space and much less converged in comparison to the controls. And the question is whether we looked at the trajectory. So if you look, I think this is how I understand the question. If you look over time, how does the movement look towards the area of the adults? And the answer is we did. It's fascinating. My student, Yosef Prat, my former student, now a PhD, created these beautiful movies where you can see. And I can't say much about the trajectories. If you're interested in exploring them, we can find them. I don't think we even published them, but it looks much more erratic. So the control group moves directly, steadily towards the adult region, while the isolated pups seem to be moving all over the place. And at the end, after a year, we studied them for a year, in many cases, they don't reach the adult zone at all. I see I have more questions in the Q&A, right? I don't know, do I have more time? Or maybe not. This is already answered, right? Yeah, I think I answered this already. I think I answered everything, correct?
Well, do we have any other questions? Yeah, there is also the Discord channel, so you can also type questions for all the speakers or general discussions. So I guess we'll have a break now, 15 minutes, and then we will return to the last session with 2 talks about NLP and early human learning.